Good afternoon, everyone. So I'm Yuta Kurashina, Research Associate in Keio University. So today's we hold a Cell Lab seminar. The today's topic is biomechanical technology, fusion of engineering and medical science. So I'd like to talk about our organization, Cell Lab. What is Cell Lab? Cell Lab is the abbreviation of Collaborative Evaluation Laboratories leading the acceleration of bioengineering. I think collaboration is one of the most important things for improvement of bioengineering researches. So bioengineering research is required a lot of knowledge about engineering, biology, engineering, physical, chemistry, and blah blah. And so we need a lot of facilities, cell culture room, microscope, and absorbance spectator, and special instrument. And we need a lot of know-how, cell culture, assay method, and so on. So we want to share and collaborate about like this. So this is the image of our concept. For example, one lab has a biology and medical science knowledge, and one lab has a physics and engineering knowledge. I want to seamless to connect like these laboratories, and I want to like this. All of these laboratories we share knowledge, facilities, and know-how, and so we collaborate on like these grants and project, and so we also hold seminar, symposium, and meeting for bioengineering. So we co-hosted with this seminar with KGRI. So KGRI is abbreviation of Keio University Global Research Institute. So KGRI is a project of Keio University Global Initiative, Keio Gijuku Daigaku Super Global in MEXT. So this organization has three initiatives: longevity, security, and creativity. The first speaker is Professor James Friend. So he comes from the University of California, San Diego, and his major field of research is medical micro device, MEMS, and his special skill is ultrasonics. So James Friend visits Keio University with support of Keio University Global Initiative. So he already come here 3 times. So please come here.
Thank you, Yuta, and thanks everyone. I'm most grateful to the funding that has allowed me to continue to come and visit with you every year. Almost every time Takamura Sensei has a birthday, so congratulations yet again. So I come for his birthday. Yeah. Yoroshiku onegai shimasu. So I'd like to talk to you today about micro-nano acoustofluidics, and I could talk about this topic for hours, really, but I will just talk about a few specific things. And I'd like to begin with a bit of a story. When it comes to using acoustic waves, there is something that many people don't really appreciate. And the idea is that when you have vibration of engineering systems, you have a maximum practical particle velocity of around 1 meter per second. And when I was about the age of most of the students here, I learned this in Japan, actually, at the Tokyo Institute of Technology. And the concept is very simple. The idea is that, this is a San Diego experience, lowrider hopping. They take a car and introduce hydraulics in the front and bounce the car. And so the height of the front end of the car is about 1 meter, and the frequency is about 1 hertz. So the vibration velocity, the particle velocity, is about 1 meter per second. And if you increase the height, then the velocity, the rate at which it will bounce actually decreases. So the vibration velocity and the particle velocity always remains about 1 meter per second. So, well, that's just one engineering system. There's another engineering system you can barely see, a speaker. And this kid has placed a cat on the speaker and vibrating the speaker with the cat. And that's moving about 2 centimeters in height at 50 hertz. And again, 1 meter per second. And then Chladni figures from EFF Chladni, who developed this idea of placing dust on tympani membranes. He showed vibration patterns. Again, 1 meter per second, because the frequency is around 1,000 hertz, and the dust bounces around on the surface with an amplitude of about 100 microns. And you would say, okay, well, this is only a low frequency phenomena, perhaps. What happens at higher frequencies? Well, a couple of works that I did when I was in Japan many, many years ago now was to make linear motors using ultrasound. And these work at higher frequencies. For example, 100 kilohertz and 1 megahertz. And so this motor, for example, is 2.5 millimeters in size. And we made one that was 250 microns in size. And again, this, for example, the bolt-on was 1 megahertz, and the amplitude of vibration is 100 nanometers, 1 meter per second particle velocity. So then the question comes is what would happen if we increase the frequency really, really high? What happens when you end up with really very high frequencies when you use this maximum vibration velocity? And the one way to achieve that, to use very high frequencies, is to use surface acoustic wave devices, where we use a single crystal substrate, usually. Lithium niobate is very, very common. And we introduce an electrode, an interdigital electrode, like the digits of your fingers. We use RF frequency electric fields, and we can generate an acoustic wave that goes across the surface, sort of like a water wave that goes across the surface of the piezoelectric material. And the frequency that's possible fairly straightforwardly is about 10 megahertz to 10 gigahertz. This is considerably higher frequencies than with the other approaches. But what we end up with as a consequence is with this maximum vibration velocity of 1 meter per second, we have an amplitude of that 1 meter per second divided by the frequency. It's very, very small amplitudes. It's impossible to see, 10 nanometers to 10 picometers, but the accelerations are very, very large, 100 million meters per second squared to 100 billion meters per second squared. And so this is where it becomes interesting, is those extreme accelerations, when you introduce a fluid in the path of those accelerations, lots of interesting things happen. There's atomization at 3 different length scales. There's droplet ejection from acoustic radiation forces. There's fluid transport across the surface, and then there's also patterning on the surface as well. And this is due to two effects. One is sound. When this vibration is transmitted across the surface and comes into contact with the fluid, it generates sound in the fluid. And when the sound propagates through the fluid, it's very, very loud, over 160 decibels. The other aspect is when the sound, as it is propagating, it actually is attenuated. It decays, and that causes the formation of fluid flow through a momentum flux generation. It's called acoustic streaming. So with that, I'm going to go through a couple of examples to talk a bit about the physics, and then to turn with at least this example and talk about the medical aspects of the use of this type of technology. This is a surface acoustic wave atomizer. There's the interdigital electrode. We have a fluid reservoir here, and there's a capillary tube with a wick. And the wick carries the fluid to the surface of this lithium niobate substrate. And so a vibration goes into the fluid and causes the surface to break up. So the question is, well, what's the physics? What is actually happening in this system? When I first started talking about this work, many people told me that, it's very boring. Look up the old paper by Michael Faraday from 1831. He did this work where he generated capillary waves on a bath of mercury. What he actually did was he took a pie tin, put mercury in the pie tin, and then placed it on a chair, and then tapped the chair at a given frequency of, in this case, lowercase f. And as he tapped the chair, he looked at the surface of the mercury and found that the vibration was 1/2 of his tapping frequency by using the sun. And so he published a paper with a pi 10 full of mercury. A very, very easy paper to publish, but it was 1831. So over the years, since that time, there have been many people that have talked about the mathematics of how Faraday waves are generated, was a nonlinear mechanism, and there's a long story. So we thought, okay, well, maybe this is not so exciting to try to actually learn about what's happening. And so we used laser Doppler vibrometry to measure what's happening at the surface of the fluid. And so we have excitation at 20 megahertz right here. And so there's a response. This vertical axis is the displacement of the fluid surface. The horizontal axis is the frequency. And because laser Doppler vibrometry is an interferometric technique, then we can measure them from very low frequency, 100 hertz, to very high frequency. In this case, the paper's a bit old now, but in this case, it's up to about 30 megahertz. And so we see a 20 megahertz excitation, and there's supposed to be then, if we are tapping at 20 megahertz, so there should be a wave appearing at 10 megahertz, 1/2 that frequency. There's no wave. There's nothing there. In fact, there is no response all the way down to a fundamental mode at 200 hertz for a water droplet about 2 millimeters in diameter sitting on the surface of this device. So you can imagine we're exciting at 20 megahertz and you see vibration at 200 hertz. It's a very bizarre kind of phenomenon. So then as we increase the power, notice that the acceleration now is 60 million meters per second squared. The amplitude of vibration is only 4 nanometers, and that's because the frequency is 20 megahertz. Then we see the improvement of the excitation frequency, no response, and then this becomes a continuous response from low frequencies to high frequencies. And there's a long story as to what is happening here, but essentially this is exciting directly the fundamental mode of vibration through turbulence. So as the sound is going into the fluid, at this very small fluidic scale, it's generating turbulent flow. And because of the extreme acceleration, we get turbulence, and it allows the generation of the fundamental mode. That fundamental mode then generates first harmonic. The first harmonic vibration and the fundamental mode combine together to generate the third and fifth. The fifth and fundamental generate the seventh and ninth, and so on and so forth until all of the modes disappear. There is no more modes existing in the system. It is a continuous distribution of vibration, and this is what's called strongly turbulent behavior. So, well, maybe this is due to the surface acoustic wave device, and it turns out it's not. If you use a thickness mode atomizer, for example, at Tokyo Hens, you know, the 2000円ぐらいあるのTokyo Hensのやつですよね. The atomizers, those work at 500 kilohertz to 1 megahertz in a different vibration mode. It's the same phenomenon. When you see the excitation here, you have superharmonics, not harmonics, but superharmonics. There's a difference. And then we have this nonlinear response, a strongly turbulent response. So it's rather peculiar. Turns out that this response from low frequencies to about 1 megahertz, here's the green line, where we're exciting now at 7 megahertz, and we see this response with no modes. What happens when you start to see atomization, droplets coming from the surface, then there's a change. It goes from this black line, green line, to this black line. There is a hump here that appears for DI water. And for isopropyl alcohol, we have another hump. Sorry, the red line here, there's another sort of hump. In the blue, there is a discrepancy. So there is, without atomization, you have a very smooth curve. With atomization, you get this discrepancy. And we suppose that maybe this has to do with the droplet generation rate, which so far no one can measure. So we try different power levels. So if you take water from 100 milliwatts to 200 milliwatts, for example, and the atomization is occurring, but this discrepancy moves location in frequency, so it goes to higher frequencies. The rate of droplet generation actually increases with increasing power. For isopropyl alcohol, it does the same thing. Triethylene glycol does the same thing. So this always appears regardless of the fluid choice, and you might wonder, well, okay, well, what is good about this? Well, there's a Kelvin, or Lord Kelvin, actually came up with an equation where he related the wavelength of the capillary waves present on the surface of a fluid to the frequency of excitation. So the frequency of the capillary wave is related back to the wavelength of capillary waves. So for example, deep water waves, you see the water waves in the ocean, those occur at a particular frequency. And so in this case, we have the surface tension, we have the frequency, we have the fluid density, and then the wavelength, and this is to the 1/3 power. The problem has always been determining what the critical frequency is. If we use this frequency of these humps, then we actually find that using this as a critical wavelength, we can estimate what the droplet flow rate is going to be. The size of droplets generated from each of these capillary waves is about the same size as 1/2 the capillary wavelength. So about 1/2 the capillary wavelength would be our droplet size diameter. If your droplet flow rate then is basically that volume of that capillary wave times the frequency again, then we end up with the flow rate versus frequency, and we actually can predict the flow rate for atomization. And what we end up with is being able to predict over a wide variety of fluids, essentially when it is going to atomize, and then how much it will atomize when it starts to atomize. So what we've done here is we've defined an atomization Reynolds number. It's not the normal Reynolds number, it's actually changed somewhat, where we use the acoustic particle velocity, like that 1 meter per second vibration, the particle velocity. We measure what that would be on the surface of the fluid, the capillary wave. And we use the viscosity, the density, and then the length scale, the size of the drop that we start with. And if this Reynolds number goes above 40, we start to see atomization. And as you increase the power on the atomizer, for example, it will go from below 40 to above 40. For different fluids, you will be in different locations. So very, very viscous fluids are very, very difficult to atomize. Well, it's because the Reynolds number, this atomization Reynolds number with a very, very large viscosity remains small. And things like methanol has a very low viscosity, very easy to nebulize, and produce high rates of flow. So one of the other problems with these devices is that they require a lot of power. The Tokyo Hands nebulizers, you have to plug into a USB, and those are about 10 watts. The lithium niobate solid atomizers are 3 to 5 watts. What we want to do is to make a battery-powered atomizer that you can take with you, put in your pocket, and then inhale drugs. But that's 3 to 5 watts is too much. So can we do better? When we looked at all the different ways to make vibration, we looked at PZT, and lithium niobate, and different cuts of lithium niobate. So for example, 36-degree Y rotated lithium niobate, 128-degree rotated lithium niobate. The thickness mode means just a simple mode of vibration like the Tokyo Hands device, or a Lamb wave, or a Rayleigh surface acoustic wave. And we looked at the quality factor and the electromechanical coupling coefficient and computed a figure of merit. It turns out that the Rayleigh surface acoustic wave, the old SAW, is really not very good. The PZT is okay. The larger this number, the better the result is. The Lamb wave devices are not so good either. It turns out that this 128 cut lithium niobate with the thickness mode produces the best value, and it's actually up here. It produces the greatest substrate displacement, over twice what you would have with PZT. So that allows us actually to get an order of magnitude increase in the atomization efficiency for our atomizers. And we have small devices with little battery-powered nebulizers, and we have one now that's about the size of a laser pointer that we can use for atomization of even very, very viscous fluids. So the question is what good is this? Why would you care about atomization and the physics of atomization? One of the chemicals we used was triethylene glycol, TEG. And TEG in the 1940s was found to be a disinfectant. It kills bacteria, fungus, viruses very effectively. The problem has always been it is impossible to actually use TEG for nebulization. You can't atomize, it's too viscous. And so now it's possible to atomize an effective disinfectant that's lung safe. You can actually inhale it. And it is so safe that you can inhale it as a patient in a hospital. So it may be possible to have these types of devices in a hospital room to disinfect the room with a patient in the room. We also are looking at drug delivery. How can you actually deliver drugs into the lungs with inhalation rather than injection? For example, one of the older studies that we did was plasmid DNA, where we were looking for a way to do gene therapy for vaccination. The idea is that, for example, H1N1, Solomon Islands influenza, the influenza vaccine normally is a killed influenza. You take the virus, you replicate it, and then you destroy it through heating. And then you inject that dead virus into humans for the immune system to use. The problem is that it takes a long time, 3 months, to produce enough vaccine. It also has to be protected with a cold chain, meaning you have to put the vaccine in a refrigerator all the way until you use it. So in Africa, they have to keep it cold, and that's not so easy. In this case, the gene therapy approach needs no refrigerator. And the other aspect about the plasmid DNA is if we can detect, we can figure out an effective gene for teaching the immune system what's happening with the virus to actually detect the virus later, that gene can be then used in this system and without requiring cold chain, and you could do so very quickly. And in about 2 weeks, we're able to produce a plasmid DNA-based genetic therapy-based vaccine for influenza, for malaria, and for other diseases. So we went through, with this particular project, we went through mouse trials and rat trials. We looked at IgG and IgA titrations, and a variety of other things to see what's actually happening with this plasmid DNA. In fact, you see the supercoiled form here. The open coiled form is here, where the open coil form is a supercoiled DNA that has had one side actually broken, and so it uncoils on the surface. And the supercoiled is the ideal form of the plasmid DNA. The open coiled form is effective, but it is one step away from being completely cut, which then becomes ineffective as a vaccine. So when we put this into sheep, and we did this work in Australia, where there are many, many sheep, and so the sheep actually like to relax, and they allow you to put tubes in their nose and intubate them for ventilation. They're very, very easy animals to work with. And so we used surface acoustic wave atomizers to atomize these very large molecules, around 50 kilodaltons to 500 kilodaltons, and atomize them for inhalation into the lung. It turns out that the very high frequency of the surface acoustic wave devices allows us to do atomization without destroying the molecules. And so we can actually do drug delivery very, very quickly and safely. So we looked at things like the Hemosiderin staining and showed that the plasmid DNA is not taken up by the macrophages. The plasmid DNA is taken up by the lung tissue and goes through the small junctures of the alveoli, the cells in the alveoli, the tight junctures in the alveoli. And we did things like the Hemagglutination inhibition assay, which is the gold standard for looking at whether the immune system has formed a sufficient response as a consequence of the vaccination attempt. And what we see, for example, in this case, these are the mice, rats. These naive rats, Sprague-Dawley rats, before vaccination, and subsequently we have a pretty strong response. We have titrations out to 1 to 1600, and after 5 weeks, we still see the same response. WHO, World Health Organization standards for vaccination is that if you titration of the blood out to 1 to 40, you still have positive results from hemagglutination, then that animal is said to be vaccinated. In this case, we have all the way out to 1 to 1600. We repeated this with a few sheep. We're only showing 4 here. And what we see is that, again, they're vaccinated. This is the first time that there has been any data at all showing that a large animal can be vaccinated with a pulmonary route via the gene therapy approach and without any protection of the plasmid DNA. Normally, you would use things like interferon or PEG, pegylation, that type of thing to protect your delivery item when you do the inhalation. In this case, you don't need to. The plasmid DNA survives all the way through to be effective as a vaccine. There's also other things that we have looked at atomization of monoclonal antibodies raised against epidermal growth factor receptor. EGFR is overexpressed on the surface of cells that tend to be more cancerous. So, for example, lung tumors tend to have cells that express more EGFR than other cells that are not cancerous. And so if you were able to detect that and knock down the EGFR activity, you may be able to slow the growth of the tumors. And in this case, what we were able to find is that these monoclonal antibodies that we raised, they're very, very complex molecules, they're large, they possibly could be destroyed by just simply atomizing. That's why most of the time they're injected. You cannot buy monoclonal antibodies as a pill to take. You can't inhale them or other types of drug delivery techniques. We tried with this and used this with a AF549 lung cancer cell line, and then, of course, the HeLa cells, the cervical cancer cells, and checked to see what's actually happening. And in fact, what we found is that with the untreated cells, we have no fluorescence that would indicate that we have no binding of monoclonal antibodies because we have no monoclonal antibodies. Then once we try the pre-atomization for the monoclonal antibodies that we raised against the EGFR, we see that we have all of them quite nicely fluorescent, so nearly 100% effective for this binding for this monoclonal antibody. After atomization, we have nearly the same result with HeLa cells and likewise with A549 cells. So the act of atomization allows us still to use these monoclonal antibodies. It's a possible route to treat lung cancer, is what I'm suggesting. Last thing is to look at human amnion-derived epithelial stem cells for clinical therapy. The idea is that when a baby is born, you have the amnion, the bag, if you will, and those actually contain stem cells. And in certain hospitals in the US, and especially now in Australia, where you can pay the hospital to keep this tissue for you, freeze it, and as the baby grows to an adult, maybe they have problems, there's some sort of disease, there's some sort of issue later, that tissue becomes available for them later. And it is therefore autologous. They can be used without risk of immune system rejection. So what we're interested in looking at is what happens to the lungs of a baby that is born prematurely. The number one problem with premature birth is underdeveloped lungs. When the lungs are born, you see the tissue of the lung is thicker. That's because it has scars. And there are many prematurely born babies that spend the rest of their life on oxygen therapy. So to overcome this inadequate lung tissue, and they never recover. Our idea is to treat them with human amnion-derived stem cells so that the stem cells actually help the lung tissue recover. It has been shown with massive doses of stem cells delivered via intravenous injection, an IV, that we have effective lung treatment where they become nearly the same as a normal baby's lung. But the problem is that it is a massive dose. Only 0.1% of the stem cells actually go to the lung from injection. 99.9% of the stem cells end up in the spleen. And so then when they end up in the spleen, they're actually useless. So we're talking about perhaps atomizing the stem cells to have an inhaled delivery to the other side of the lung tissue as opposed to going into the blood route side. So can you atomize stem cells is the question. And it turns out that you can. And this is the calcine stain. Green is good, red is dead. So the calcine stain shows that atomized stem cells are nearly identical and alive as the control stem cells, all of them green, no red. We also looked at the immunophenotypic profile at 3 days via assay, and we were looking at, these are murine stem cells, and so we were looking at CD29, 44, and 106, and essentially we have a negative control where at the left of the line, if the FITC is weak, then we know that we are not seeing what we want to see, and these are to the right. Whether they atomize or not, essentially the results are the same. So the CD29 in this case is being expressed in exactly the same way. We may actually quantify this. Then what we will see is that the CD29, 44, and 106, after 1 day post-atomization, shows a bit of a discrepancy, particularly CD106, compared to the unatomized control. If this bar was at 1, then that would be said to be equivalent in the genetic expression of CD106 to the unatomized stem cells. 44 is nearly the same, but whatever the case, after 3 days, everything is back to statistically no difference post-atomization. Okay, so those are some of the medical applications of atomization. I wanted to change direction slightly to talk about something a little different for the last bit of time that I have. It's acoustic nanoslits. With a lot of the work that has been done with acoustic waves and ultrasonics and so forth, it's all at the micro scale. There's a lot of ideas of doing microfluidics, not so much with nanofluidics. And so we wanted to take a look at some of these things, where what we do is we take one of these surface acoustic wave devices and generate acoustic waves across the surface and place upon it another piece of lithium niobate that's bonded to the surface. And that lithium niobate actually has a nanoslit, a channel of width of about 10 microns, length 4 centimeters, and a height, in this case, it shows about 100 nanometers, and our latest devices are 5 nanometers. And then we place a fluid into that nanoslit. So the process is straightforward but annoying. So making the surface acoustic wave device is very, very easy. It's a photomask of ultraviolet lithography, produces your metal electrode on the lithium niobate substrate. Making the other side, the channel, is not too difficult either. You have a mask, you form a metal mask, and then you etch with deeper active ion etching. The etching time of which from 0 minutes out to 10 minutes, you can get a channel depth in this arrangement down to around 10 nanometers. And we've changed the profile so we can get to 5 nanometers now. And the bottom of this is very, very flat. It's a nice feature of deeper active ion etching, is it produces very flat profiles. The problem is this part, bonding lithium niobate to itself. It turns out the lithium niobate is really a challenge to bond, and we found a room temperature method for bonding where we use a complicated cleaning process, but it's not hard, it's just tedious. Many, many steps. So let's summarize here with the 4 steps to actually make it possible to do. And here's what we have. This is the sort of dimensions. You can see here's the nanoslit. So it goes on for a very, very long time. and it's also 4 centimeters deep, but the height is very, very short. So you might say, well, what would you actually do with this? Well, that was sort of a question of what actually happens. It was the first reason we try to do something like this. And this, for example, this is an empty nanoslit, and we have 2 holes. And we place fluid, water in this case, in this hole, and it will begin to fill, because lithium niobate is hydrophilic. It likes water. And so over time, the water fills from right to left. So 0.2 seconds, 2 seconds, and 4 seconds is completely filled. It fills according to Washburn filling, meaning that at first it goes very, very fast. So it moves very quickly, this meniscus, X position from right-hand side, so X is 0, X is a little bit larger, larger, larger. The X position of the meniscus goes with the square root of time. The velocity is fast at first, but it slows down because of viscosity. As a fluid moves in that thin channel, slows the fluid. The Washburn theory predicts a very, very rapid meniscus advance. The experiment is slower. The reason it's slower is because Washburn, when he did his experiments in the 1930s, he looked at microchannels and millichannels. And there was air in his channels, but air doesn't really matter in his case. At this scale, air is very important. It's actually difficult to push air through this channel because it's so very thin. When we look at using surface acoustic waves, we actually speed up the response, but it's not remarkably sped up. So I thought, well, maybe this is a disaster in experiments. And so the reason I show this is because this is sometimes what happens with research, the panic. And we looked at some of the analysis first. So for example, the Washburn position of the meniscus with respect to the square root of time also depends upon the surface tension, the wettability, and the viscosity of the fluid. And we can predict a similar sort of meniscus advance from the acoustic wave and add these together. This position of the meniscus for the surface acoustic wave depends upon the vibration amplitude as a ratio, and then the vibration velocity squared. There's density, viscosity, and then the frequency in there as well, but those are in the square root. This simple equation is actually obtained through rather complicated analysis, intermediate asymptotics. And so there's a lot of analysis. Many people working in this discipline choose to use COMSOL or some sort of analysis. You don't have to, but this is a little bit painful, but it's worth it. So if we add these two together, we can get an approximation for how fast the fluid fills the channel. And in fact, there's the theoretical meniscus position, and this is the experiment. If all the data was on this line, then we would have exactly the right result. When the experiment was at 2, the theory would be at 2 also. And when it's at 7, it would be at 7. But in fact, the theory says that the meniscus is faster than it really is. Why? It's because there's air, and the air slows down the filling of that channel with the water. What's really interesting is what happens when the acoustic wave and the fluid are in opposite directions. In this case, we have the filling from right to left in this 100 nanometer tall channel. There's the empty, shows as red because of the interaction with light. And so the fluid is filled over to here. We turn on surface acoustic wave from left to right, it pushes the fluid back out. Only reason that matters is that this water really likes lithium niobate. The capillary pressure to fill the channel is 1 megapascal, 10 atmospheres. So to push the fluid out, we must generate 10 atmospheres of pressure from the acoustic wave to push the fluid out of the system. And so there's a lot of questions as to what's actually happening. Why would you actually get this? Is this standard old acoustic streaming that's causing this? And when we published this first paper, we thought it was just acoustic streaming. It turns out that it's not. That some calculations shows pretty easily that we would need 1,000 more, we'd need a factor of 1,000 increase in what is happening to get the pressures we are able to generate in the system. So we had to go back and do more analysis to figure out what is actually happening. Turns out that there's a nonlinearity here. The vibration of the channel as the acoustic wave goes by is about 10 nanometers in amplitude. Even though it's at 10 megahertz frequencies, the amplitude is at 10 megahertz, and the height of the channel is about 100 nanometers. So what we end up with, the leading order instantaneous pressure with this case, because of the constrained system, is 1 megapascal. That's what's responsible, is that we're squeezing the fluid in this narrow region between two surfaces, one of which is vibrating at a significant amplitude compared to the thickness of the channel. This, though, notice the sign here, that averages out to 0. Over time, the pressure is positive and the pressure is negative. Pressure is positive and it averages out to 0. The reason that we get a non-zero result is because if you look at the flow rate, Q, there is an h cubed here. This also averages out to 0, except that this h cubed, okay, depends upon the height of the channel without saw, and also this part, the induced displacement of 10 nanometer amplitude. If we cube h here, and then expand this term, let's show you what happens. So we have h cubed is the height of the channel cubed plus 3h squared times all of this stuff. This doesn't make any difference because it's constant. But this cosine multiplies with that cosine. If I average that out over time, that's cosine squared averaged out over time, and you end up with a non-zero average. We just made a pump, an acoustic pump. And this is a different kind of acoustic streaming. That's what's been shown before. It's a peristaltically induced streaming. So it's rather different than some of the past material. When we look at wider channels, they show very weird drainage processes. So we have these de-wetting regions. This green is actually fluorescent from the water that's been dyed with Fitzy. And we see de-wetting. And the de-wetting regions combine together, and you end up with these islands left behind. All of these structures about 1/2 the size of the wavelength of sound in the fluid. One accidental experimental result, and for those students that I would encourage you to, even the mistakes sometimes are very, very important. This is an example of a mistake. Because the student, Morteza, he made a mistake of filling the channel, and instead of filling the channel completely, he ended up with a droplet. And when we did the calculations for the size of the droplet, we realized it's somewhere between 1 and 10 femtoliters of water. That droplet stays in this system for 30 minutes. It doesn't evaporate. If you place a 10 femtoliter droplet of water on the lab bench, it'll evaporate in a few milliseconds. But in this system, because of the closed-in nanochannel, then it will last for 30 minutes or longer. What we found is that if we make a T-shaped channel that has a change in width, then this has slightly greater capillary pressure, 600 pascals only. So the water wants to fill into the system with 1 megapascal of capillary pressure and go from here, the wide part, to the narrow part with 600 pascals. That's enough to cause a fluid droplet to go over to the narrow region. If we turn the surface acoustic wave on, we can push the fluid out. You turn it off, it'll go back in. And what it suggests is a way to actually make a droplet-controlled system by having undulating lateral dimensions, having narrow regions where you trap droplets and wide regions where they don't, where they're not trapped. And you use surface acoustic wave to actually shuttle them around. And this is about 3 orders of magnitude smaller than any of the other sort of results that have looked at these sort of phenomena. Last thing I want to mention is it's possible because of the fluid pumping characteristics with a 1 megapascal pump, you actually can continuously pump fluid through the system. We can go through a 10 microliter droplet of water, for example, in about 1 minute. And so it very rapidly flows fluid through. With a 1 megapascal pressure, it will drag everything through the system as long as the particles and objects are smaller than the height of the fluid channel. If they're larger, they get trapped at the entrance. So for example, siRNA loves to be agglomerated. If you buy siRNA from Sigma Aldrich or a company, it comes in clumps. Those clumps are much larger than 100 nanometers. what's possible to do is to take a droplet and then draw it through, and you end up with individual siRNA molecules in that nanoslit through deagglomeration because of the very high pressures. So I'm going to stop there, and I wish to acknowledge my great group. They make me look good. I'm an idiot, but they're really awesome. And all of funding, of course, and the collaboration with people here, particularly Professor Takamura. Thank you.
Thank you for sharing your valuable presentation. We have a time for some questions. Please raise your hand. Okay, so.
Yeah, thank you very much for your nice talk. So I'm very interested in many topics in your presentation. So, okay, so I have a question about stem cell delivery for your nebulization with vibration. So it means you set the dispersion of stem cell suspension of stem cells on your device. Then the cell is encapsulated and split. So then I'm very surprised that the cell is alive.
As am I. As am I.
Yeah. So why? So I heard that, for example, homogenization or supersonification breaks the cell structure. But
yes.
If you apply this very strong energy to the fluid, but no damages in the cell, so why does it occur?
The reason is two reasons. One of the main reasons for the main mechanisms of damage of molecules, cells, large objects from ultrasound is cavitation.
Cavitation. Okay.
Yeah, and the propensity to have onset of cavitation, the ability to have cavitation, goes down with the square of frequency. So if I double the frequency, you must quadruple the input power to have the same cavitation. And so at about 1 megahertz, it becomes very, very difficult. So you need about 1 kilowatt. And above 1 megahertz, then progressively it becomes more difficult from that point. Okay. In fact, in this system, above about 5 megahertz, it is close to 10 kilowatts, and we use less than a watt.
Okay.
So that's cavitation is suppressed.
Okay.
There's one reason.
So so you mean the fluid shear stress doesn't affect, not.
That's correct.
Just only the cavitation may causes a kind of.
The cavitation is one significant contributor, and the ability to generate cavitation depends upon the displacement amplitude. And that displacement amplitude goes down significantly with frequency. Okay. Yeah.
Right.
The other reason is shear stress.
Shear stress.
But the shear stress is only effective on molecules over a narrow frequency range, about 10 kilohertz to 100 kilohertz. Outside that frequency range, then the shear can be very, very large, and it doesn't affect the molecules.
Okay, thank you.
So any questions?
Thank you very much for the nice talk. I'm also interested in about the application of nebulization. I'm experimentalist, so I'm very interested in what should we set up to apply the technique to perform the experiments. What kind of equipment we need to do?
The atomization, the basic atomization is really very easy. You need a signal generator, amplifier, and piezoelectric element, and that's about it. Yeah. But some of these devices, if you wish to do the measurements, the vibration of the acoustic wave or the amplitudes, then you need a fast measurement technique. So I use interferometry, but there are others, I guess, to do that, mostly based on lasers, though. Depends upon the device. The resonant frequency of the device will help you narrow the range. But the problem is that even if you choose a frequency, the atomization frequency and phenomena is totally different. So, for example, we have made atomizers from 1 megahertz to 900 megahertz And almost always it produces the same droplet size. Yes, yeah. The old theories from 1960s say the droplet size should change based upon the frequency to the 2/3 power, but it doesn't. It doesn't. It's because the physics it's not a Faraday wave. It's actually strongly nonlinear, turbulent acoustic coupling. This bizarre phenomena. Yeah. My pleasure.
So any questions?
May I ask about the device? Yeah, in the later research result, in the second half of your presentation, you put a very in nanometer size flow channel part.
Yeah,
You put that part onto the SAW device, right?
that's right.
So if that's the case, it is easy for the wave to be reflected at
Absolutely.
when it is contact with the channel. So what is happening inside the device at that time?
Yeah, a good question. This is a little deceptive. The channel that we make is wider than the aperture of the SAW device. So what the SAW sees is only the fluid, but then of course the acoustic wave goes up, and there is an acoustic wave generated in the superstrate also. That also is SAW. So there is an acoustic wave on the top and bottom of the channel. The top channel, that amplitude of that acoustic wave is about 30% of the amplitude of the acoustic wave on the bottom side. The fluid itself, there is actually no sound generated in the channel when the channel height is below, in these systems, about 250 nanometers. So you can show that there is actually no sound at all. It's impossible. But because the two walls, the top and bottom wall, are vibrating with sound, then it causes lateral responses. That's one of the reasons why. Sorry. We see such bizarre responses. Is there a lateral coupling, and it's due to the vibration in the solid materials, both top and bottom.
So the figure of this figure should be changed because due to the height of the channel, right?
That's right. Okay. That's right. Although almost always the scale, of the features is 1/2 the wavelength of fluid. Even though there's no sound, it's probably getting vertically. It's a little bit strange. Yeah.
Okay, so it's on time. So everyone, once again, please give Professor Friend a round of applause.